Yeah, that's what I was going to ask. I mean, this was new stuff at the time. This
Oh,
was new stuff for the
incredibly new
FDA to
stuff.
consider.
It
Yeah.
was. It was. This was 1988, 89. So
Hmm.
very new for the agency. The combination drug device pathway was still very, very new, which, we've come a long way. Last year, over 400, I believe, combination drug device products were approved by the FDA. So they're it's a well-paved road now. But back then it was the Wild West. We were making it up as we went along.
Yeah.
So the CO2 ended up needing to be developed as a drug, and it was an unfortunate reality that commercially no one was going to pay pharmaceutical margins on a gas that you can get in virtually unlimited supply. I mean, I'm producing a lot of CO2 right now for nothing.
Hmm.
Nobody's going to pay pharmaceutical margins prices for that. So commercially, the project became a non-starter, and we negotiated with the agency, and we ended up coming to the realization we had to come up with another way of delivering CO2 that could fit in the device world, which we ended up doing a hand system that got by. But everywhere outside the United States, our CO2 system was commercially successful.
Hmm.
So we sold hundreds and hundreds of those systems, and CO2 angiography is a kind of a standard of care these days. In the U.S., it's still done by hand, but in the rest of the world, it's done with a combination drug device system.
Yeah.
That that got me very, very well cemented into drug device kind of being my thing, and more opportunities started to come around, and I took my team and I took AngioDynamics public in 2004. We had a great run. We were the best performing medtech IPO that year. And we were Forbes top 200 growth company. We were a Business Week top 50 hot growth company, and we were just crushing it. I ran it as a public company for 5 years, which is about the average for public company CEOs
Yeah.
shelf life, and then was promoted to co-chairman in 2009, which is code for you're out of here. We're So the board thought it was the time to bring in a Fortune 500 level CEO to really drive the company. It was an extremely successful company. On my watch, we got it to over $1 billion in market cap, and it was a great return for the shareholders, great return for my original investors at EZM, and life was very, very good. So that was 2009, and I had to decide what I was going to do with myself. So I decided I'd join a bunch of boards and try and give back, frankly, to young entrepreneurs. And one of the companies I joined was Delcath, that had a very interesting combination product system in oncology. And many of the products at AngioDynamics ended up being in oncology
Mm.
because the interventional radiology community was migrating that way, minimally invasive, focal oncologic therapies. So I had a good base in oncology, and the Delcath clinical data. They were phase 3, developing a ocular melanoma mets to the liver new therapy, which was a drug device combination where the vasculature of the liver was isolated, and the liver was exposed to very, very high concentrations of melphalan, one of the original cytotoxic chemotherapeutics.
Hmm.
And it would kill the cancer. And amazingly, the healthy hepatocytes, the healthy liver cells, were virtually immune to the chemotherapy. And when you think about it, the liver filters poisons out of our blood. So it made sense that the hepatocytes be able to tolerate this. If the chemo got out of the liver, it would decimate the bone marrow and lead to death, or at least neutropenia or thrombocytopenia, or both, grade 3, 4, really, really toxic. So it was very important that the device do its thing. And it involved a high flow blood filtration system that would filter the chemo out without damaging the blood, which had never been done before. So it was a really intriguing project and required a lot of innovation. And these patients were terminal without an innovation. So it was incredibly gratifying to be able to add, significant quality of life to these patients' lives where they really had nothing that was working before.
Yeah.
So a belly full of that, we published our interim pivotal phase 3 data. It showed incredible efficacy, and the company's market cap went to well over $1 billion, about 1.4 billion. It was 28 million when I joined the company. And we thought we were cooking with gas. We filed our NDA for the combination product and got a complete response letter, which was a bit shocking, where they said, "Hey, we love your data. It's really great, but you changed the device during the phase 3 trial. And we want you to repeat the study with the same device. We also, your study design included a crossover provision that allowed patients who failed the standard of care, which they all failed, to cross over and get the therapy if they still qualified, which was ethically, a very reasonable thing to do. But unfortunately, on an intent to treat basis, that meant the Kaplan-Meier curves were overlapping because the patients that crossed over did just as well. So there was no difference between the control arm and the treatment arm when you accounted for the crossover. When you subtracted the crossover compared to historicals, there were, first off, all the crossover control arm failed to get there. Two, if you compared it to any historical data, all the patients died. So it seemed really harsh that the agency was throwing the crossover provision in our face. But during the course of the execution of that trial, the FDA had made a big shift and said the crossovers really, although they're ethically attractive, they really do a disservice to getting the new therapy approved. You really need clean data. So you've got to randomize people and not cross them over. So we suffered through that. Delcat, at that point, we were looking at having to repeat the trial. It was an 8-year process. I handed the reins over to my second in command, Jennifer Simpson, who ultimately ended up getting it approved last year, her and her team. So great success. But I went on from there to another pharma company that I was on the board of that called Antares Pharma, which made combination products. Auto-injectors and bio-betters, taking existing drugs and making them better by applying a device technology or a transcutaneous delivery technology or a depot formulation, really, truly bio-better. Mm-hmm. And it was quite a factory. It worked with all the big pharma companies and developed very, very innovative and proprietary technology for them and would manufacture the products for them. So I got a real operational education there in development, in CMC, in device, a big device division in Minneapolis, Plymouth, Michigan. And it was really fantastic. I took the job as a director going to CEO because the CEO had left at a very tough moment. There's never a good moment to take a marvelous opportunity. But it was sort of a tag you're it at the board level. Eamon, you're the best guy and capable guy to do this, and would you consider doing it? And I thought the number 2 there, Bob Apple, was the obvious choice, and I said so. The chairman knew Bob Apple, had raised him from a pup, as he said, and said, "No, I don't think Bob's ready. I love him like a son, but I don't think he's ready." And he knew him better than I did. But I thought to myself, if he's not ready, neither am I. He looks pretty good to me. And so I took the job on the condition that everyone would agree I could develop Bob, and I was quite confident I'd be able to do that. Turned out that was one of the hardest jobs I ever had because when you're CEO, you're the boss. You get to do what you want to do, and you own it. If you're right, you own it. You're wrong, you own it. And life's pretty simple. When you're as a CEO who's developing an imminent number 2 to take over, you're really always trying to get them to do it and do it in a way you can live with. And Bob Bob's his own man and quite talented fella. And his way of doing it wasn't always what I would have done. I told him, and we're very good friends and colleagues, and but it was really hard to kind of be that mentor. Because at the end of the day, in a public company, you're still responsible. So it was a little more stressful than I thought at the time, but it all worked out great. Passed the baton to Bob in 18 months, and he went on to have a great exit with Antares Pharma. It was acquired a couple of years ago for a couple of billion, so it was all good, and very proud of him. He did a great job. So when I left Antares, I said, okay, I'm done with the CEO thing. Enough is enough. I'm going to find something that really is what I want to do without being a public company CEO. So I ended up at the Brigham and Women's Hospital, Harvard Med School. They brought me in to get their translational medicine accelerator off the ground. They had gotten a big endowment to really fund translational medicine accelerator, really going from bench to bedside. And they said, we want an entrepreneur like you who has drug and device development experience to mentor our faculty, teach them how to be entrepreneurs. And I thought, wow, this is cool. I'll have my arms around the whole pipeline of Harvard Medical School. Imagine the faculty at the Brigham. I mean, the best and brightest, Nobel laureates, et cetera, et cetera. I said, this is crazy good. And it was absolutely magic. And the faculty was incredibly appreciative, even when my lesson was, your baby's ugly. And you need to kill it, fail fast. They were still appreciative that they learned a whole lot. And I thought I was done. I was really very, very, very happy there. And I got a call from a former colleague who, a research physician, who said, hey, Hobbs, you have to come see my data. I'm curing metastatic prostate cancer. And I said, buddy, I'm literally standing in the hallway at Dana-Farber, and nobody's curing metastatic prostate cancer here. Yeah. So how the heck are you doing that in Fort Lauderdale, of all places? So he said, well, come down, I'll show you. So I did, and he did. He had developed, or helped to develop, a new method of immunotherapy that was showing incredible promise. And in fact, he had a case series where he was very successfully treating late-stage castrate-resistant prostate cancer patients who were hospice-bound and getting complete responses. Unheard of, including resolution of bone mets. As I peeled that onion, it became clear that he had adapted his case series from another physician who I knew, who had trained him, who had over 400 cases at the time, by the name of Jason Williams, who's the real pioneer of this Sync-T therapy. Mm-hmm. So went to Jason Williams, did a deep dive on his data. Most of it was in metastatic lung and breast, a combination product of a complex immunotherapeutic with a new device. And I said, wow, this is a massive breakthrough. If it's real, it's anecdotal data, and well, boy, it sure looks real. So I brought in a team to really go through the data, including my old friend Chuck Klink, who was a year behind me at the Air Force Academy. He didn't finish either. He went on to med school in the middle of the Air Force Academy, but in the small world department. And Chuck's a medical oncologist, NCI trained, NCI fellow, and an entrepreneur himself in immuno-oncology. He came in, peeled the onion, he said, Eamon, this is the best thing I've ever seen. We need to figure out how to get this to patients. And I said, I'm thrilled to hear that. I'm all in. So we put together Syncromune and funded it, and got a phase 1 trial in prostate cancer to the point where we could publish interim data, which we did at the American Association of Cancer Research in April. And I delivered again at ASCO one-on-one with some of the leadership, or every leader we could hold still.
Yeah.
And and it's just incredible data. We have an 85% in our evaluable patients, which is 13 out of the 15 in the trial at that time. 85% overall response rate, and 5 complete responses, which included complete resolution of bone mets. In immunotherapy, when you get a deep, complete response like that, it's very often durable, and after 5 years, you can start to call that a cure. Our longest patient out now is well over a year, disease-free, and we couldn't be more thrilled. We have another phase 1 trial with a different drug in metastatic lung and metastatic breast, and we just had I can't talk about that, but we're having great success there too. Really exciting. It's truly the best project I've ever been involved with. It certainly has the potential to be the most impactful, in that there are millions of patients every year who can benefit from this therapy, and it's just magic. I think it's worthwhile to talk about the complexity of the drug device combination, in that it's not just a drug device combination, and those are hard. We all know that. I always say developing drugs is really hard. Developing new innovative devices is really hard. When you combine them, it's really, really hard. It's kind of the Mount Everest of challenges, from a development, a regulatory, from clinical development, everything is just harder. So the juice has got to be worth the squeeze. The first thing to think about drug device combinations they are going to be harder. But if the juice is worth the squeeze, so what? We know how to get these over the finish line. You just need the resources to be able to do that, which can become quite significant. So if the market's big enough and the unmet need is big enough, then it's still very viable. And in this case, that's the case. So SyncT is a combination of a proprietary device that creates an in situ vaccine by doing oncolysis of a small part of one selected tumor. Mm-hmm. We're not trying to ablate the tumor, not trying to kill it. We're just trying to rupture cell membranes in a preponderance of cells in a small part of the tumor to release the cellular contents, which include the all-important antigens, tumor antigens, which the immune system can use to build an immune response to that specific cancer. So it's a personalized vaccine. And into the device, the second thing the device does is provide a conduit or a needle to infuse the drug, which is a multi-target immunotherapeutic comprised of 3 monoclonal antibody checkpoint blockade, and one nucleotide, one oligonucleotide TLR9 agonist. So we are hitting the immunosuppression with multiple targets, which is quite effective and been shown to be quite effective with systemic therapies, but prohibitively toxic, and being highly immunostimulatory at the same time. So the drug is doing a lot of things all at once. But the mechanism of action really is to synchronize, hence the name Syncromune, synchronize the location of the 3 things you need in order to get an activated and properly educated T cell. And that is, you need the antigens, the tumor antigens, you need the immune system components to do their thing and educate a T cell. And then you need the drug necessary to combat up the highly immunosuppressive aspect of what makes cancer and be highly immunostimulatory to force the interaction to take place when you've synchronized the location. So the antigens, the immune system, and the drug, that's what the infusion does. And the magic can happen in the tumor microenvironment, but we think more likely the infusion washes the antigens and the drug out of the tumor microenvironment into the draining lymphatics, where we all know have the highest concentration of immune system components in our bodies. So it's the perfect place to create the synchronization of location. So think about this. It's a brand new device that has absolutely no utility for anything. You don't want to lyse tissue in the body. You want to ablate it and a margin of healthy tissue around it to have a positive effect in focal tumor therapy. So lysing a small part of it doesn't do anything for anybody except in our therapy. Yeah. You want to infuse a drug in relatively high volume into through that conduit. That's going to take the lytic debris and wash it out into the draining lymphatics. And then you have a 4-component large molecule co-formulation. Other than that, this is really easy, right?